this afternoon we talked about an expansion of the universe in terms of the possible mechanisms of DADA2, hearing about various things such as the role of type 1 interferons and adenosine and netosis in the pathophysiology of the disease, and also an expansion perhaps in terms of the types of cells that may be involved in the pathophysiology of DADA2, looking now at the role possibly of endothelial cells intrinsically in the development of DADA2, and also the role of neutrophils. And maybe, again, just to stimulate things, and then I promise I will not ask any more questions unless provoked, is the question of endothelial cells. And so back in the olden days, we talked about the idea that really the primary locus of expression of ADA2 or CECR1 at the time was in monocytes and macrophages and other myeloid cells, and in fact had at least some data suggesting that it's not expressed in endothelial cells. And now it appears that perhaps it is. And I just thought I would throw that out as a question as to what is the relative expression of ADA2 in endothelial cells, and is there an intrinsic role in endothelial cells, or is this something that's really coming from the white cells? And I think, Dan, you had at least provided some of the data back in the original description of the disease, adducing evidence that perhaps it's not expressed in endothelial cells, at least under some conditions. Do you want to address that?
Yeah. So in our first paper, we compared the several endothelial cell lines like the HUVEC and the human dermal macrovascular cells compared with the myeloid cells, and they are very little protein, almost nothing Western blot showing the protein band, and also the activity of the ADA2 showing nothing. But we still think we just cannot just give up, still need to dig out what these endothelial cells, maybe they are not at the baseline, they maybe under the pathology conditions, they will increase some ADA2 expression. That's my thought. So I go back to the question again recently, and I test another group with HUVEC and stimulate with LPS and TNF-alpha and interferon gamma, and compare with the gene expression of CECR1. Basically, the LPS interferon TNF-alpha didn't increase anything, but the interferon gamma has a little bit increase with CECR1 mRNA expression level. And we also test the protein expression by Western. I still cannot see any detectable nice protein band in HUVEC. And right now I'm trying to compare something like different cell lines, like either lung macrovascular endothelial cells, and also during the developmental, maybe they have some change. For example, we're using the iPS-derived endothelial cells. We can compare at the early stage and the later mature stage what the ADA2 expression going. So I think that's a very good method, can kind of like mimic in vivo when these endothelial cells like develop from the beginning how this ADA2 works. So that's my hypothesis. No, okay. I don't know, really.
Does anyone else want to comment on this question?
Maybe. Different transcripts, like this your antibody does not recognize this specific transcript. I don't know if there's anything known about different.
Antibody works, we use it for myeloid cells. It just cannot. So the antibody works. We use the myeloid cells as a positive control, so they have a clearly ADA2 band. They just like very low protein expression in this endothelial line. They do have the RNA level. They do have kind of like not very low cycle. The qPCR-CQ about like 26 to 28, not very low, but the protein we cannot really see very clear detectable band.
Because in our staining in immunohistochemistry or in there you can recognize endothelial cell just as a, let's say, morphology, but we do not detect expression in tissue. In this.
Can I ask whether you looked for ADA activity in those cells, and was it inhibitable by ENA?
ADA activity?
In other words, was there ADA1 as well as, or did you look for ADA1?
Actually, I didn't look for ADA1 yet. Yeah, I probably should do, yeah.
Okay, well, why don't we open things up to the rest of the group? So Gigi, it looks like you have a comment or question.
Yeah, I found very interesting the story of lysosomal DNA's activity of ADA2. I have two questions. One is this a result of the dimer, or can a monomer also function as a DNase enzyme? And the other question is, has anybody tried a correlation, in particular your group, since you are the ones who have discovered this? Have you tried correlating that activity with the enzyme activity as measured by the other assays, or in fact do different mutants behave completely differently with the two assays?
Maybe I can start with the second question, and then I give to Ole the reply to the first one. So for the second question, what we have tried to do is to actually to produce different kind of mutants, actually 3 different kind of mutants, but differently for what, for example, it has been shown before. We managed to produce only the G47A. The other 2 mutants, they could not be expressed at all, and we could not purify, so we could not test. We tested the G47A, and G47A has a reduced, normal DNase activity, but actually in our hands it has also a normal adenosine deaminase activity. The only difference that we see is in the level of expression, and this is true in the patients or in the proteomic data, but it's also true when you express the protein in eukaryotic cells. So our cells is not bacterial cells, is produced in egg cells. And also in that condition, the level of the expression, so the amount of protein that we can purify is reduced in comparison to the wild type.
Does it depend on the activity of the enzyme, this endonuclease activity? Have you tried to use a mutant that has no AD activity in your experiment?
So this was my reply, sorry. What we tried to do is we tried to produce 3 different mutants. For 2 of them, we failed in producing the protein, so we could not test the protein, the activity, because we don't have protein. For 1 of them, we could produce the protein less efficiently, and the function in terms of DNase activity and adenosine deaminase activity was conserved.
Wait, can I ask that same question? So was that the G47A? And that mutant you were able to express, did it have ADA2 activity?
Yes.
But patients who carry that mutation don't.
They don't? But they don't have the protein also.
Is it secreted?
The G47A? Yeah. In our hands, yes, we did. We purified it and we did it secreted in overexpression, yes.
Just comment on using the 293 transfection system, that G47A gets about a 40% expression and secretion compared to control. So it's reduced but not absent. Exactly. So it corroborates your data there.
And is it active, or in other words, is what is secreted active or inactive?
It is active. It is active. The secretion is decreased by more than a half. So I think that's exactly what they're saying as well.
Yeah. That is a finding with the difference that you do not have in the patients. If you measure the activity, you don't find it because the protein is not produced. It's produced less.
Yeah, I just wanted to answer the first question here. The first thing is I want to say that finding the DNase activity was a big shock for me. That's the first thing. The second thing is if the ADA2 can function as a monomer. And that's another shock for me because it looked like when I purified the enzyme, it seems that the molecular mass is comparable to a monomer and not a dimer. So I just wonder, it's possible that the ADA2 is activated in the lysosomes in some way. But yes.
Could I do a comment from a clinical point of view, because I'm a clinician, and we listened this afternoon several data suggesting that DADA2 is a type 1 interferonopathy, and this could change the therapeutic approach in our patients, especially in the patients, the minority who don't respond to TNF inhibition, using other kind of treatment, such as JAK inhibitors or mycophenolate. Because this is a personal opinion, I think that this disease shows many features similar to monogenic lupus. If we think the autoimmunity or some patients show low levels of complements and neurological involvement and renal involvement and hypertension. And so I think that, for example, for that patient with an hematological phenotypes, before to proceed with the bone marrow transplantation, perhaps a different therapeutic approach could be done. I don't know, which is your opinion?
Well, my opinion is definitely enzyme replacement therapy, but that's used with success for several lysosomal storage disorders. I also want to say one thing about the thing about the question about secretion and transport to lysosomes, because you may just be very confused now that why does a secreted protein end up in the lysosomes? But this is a typical feature of mono-6-phosphate-dependent lysosomal proteins. It's not specific for ADA2. So all mono-6-phosphate-dependent lysosomal proteins will increase their secretion upon activation on the macrophages. And that's probably due to some kind of a regulation of the phosphotransferase that is somehow inactivated macrophages. So we are not talking about any discrepancy about the results. It's an interpretation of the results which is important.
Marco, did you want to say something?
Maybe we have to make a point on the interferonopathy because, I mean, again, from a clinical point of view, I think it's rather important, in my opinion, because indeed, if you try to treat a very trivial clinical observation, lupus patient with sting disease, or a SAVI, or a candle with anti-TNF, we make a disaster. And we know for the other and we know from the other hand that all the drugs, many of the drugs that has been used in DADA2 that are effective in lupus and the other disease are not effective in DADA2, cyclophosphamide, azathioprine, and so on. So I think this is, an important point because we have to do the best of our patient and start trying to understand why these patients respond so beautifully to especially those with inflammatory phenotype to anti-TNF. So I think this is an important, I understand it's very trivial. But when I go to of course the presentation of Dr. Grayson is much more reassuring to me on the fact on the possible role of netosis in activation of macrophage and production of TNF, and maybe reduce this interferon signature more to an epiphenomenon of abundant presence of DNA during the inflammation that you show very, very, very beautifully. And so, I mean, I think this is, to me, and I hope that you can explain to me and make some light on that, because I think it's a very relevant question from the therapeutic point of view for this patient.
Yeah, I think you make a very good point, Marco, that certainly the implications of thinking of this condition as an interferonopathy in terms of how would one approach it therapeutically, it's really a critical thing. I think that it's important in terms of understanding the biology of the disease and the molecular details of what's going on, but certainly getting to the level of treatment, we do have to think a lot about the consequences.
Because actually, I mean, this is true for AD2, but it's true also for the classical type 1 interferonopathy. So they also, I mean, also in that field, it is actually discussed how pathogenic is interferon. So we have a lot of interferon in those diseases, but is that the molecule inducing the tissue damage? This we don't know. It is discussed. I mean, many of the people in the field, they are discussing this point. There is another, for example, also in the recently discovered disease, so DNase type 2 deficiency. So recently described by Crow, they do find interferon, but they find a lot of other cytokine. For example, they find TNF into the bone marrow. Mm-hmm. So interferon is considered the prototypical, let's say, cytokine induced by nucleic acid sensing, but it's not the only one. Mm-hmm. Then there is another point that is, in my opinion, extremely important, that it differentiates ADA2 deficiency with the other interferonopathy. That is the selective expression of the protein only in monocytes that makes this disease completely different from the other interferonopathy, where the expression of the enzyme is much more, let's say.
Well, here I would like to make a comment that it is not really specific to monocytes, as we have checked various different endothelial cells. Although if you take HUVEX, which is considered as a model system to study endothelial cells, definitely they express very low ADA2, but we did check expression of ADA2 in other endothelial cells that are relevant in context of the disease pathogenesis as dermal cells or brain microvascular endothelial cells. And they do express like a lot of ADA2. So
maybe you misunderstood me. I do not say that is exclusively expressed by monocytes. I said that is preferential. So, I mean, the level of protein, if you measure in proteomic analysis, I didn't do in endothelial cells, but the level of protein is higher in monocytes. But for example, also if you analyze different kind of monocytes, the level is strongly different, even inside the monocyte subtypes.
No, again, I would say that we compared it to monocytes as well. So these different endothelial that I'm talking about, that is dermal or brain, if you just compare the cellular level, they express more than monocytes. The cell line, I would say. I'm not talking about the primary monocytes, but
which kind of endothelial cells? Excuse me? Which endothelial cells?
I said like brain and dermal microvascular endothelial.
That could be. Because different origins of endothelial cells, they could have different phenotypes. That's totally true. So I have some, sorry. I want to have some comments about this interferon, type 1 interferon signature. Because currently I'm working on both SAVI and the DADA2 disease and on these two projects. And so I think they are have very similar thing. It's, for example, STAT1. And I use the iPS-derived cells stimulated with like interferon gamma, for example, in the ADA2 cells. And you can see very significant increase with STAT1 expression. And yes, so they are kind of similar, like you're showing higher type 1 interferon signature. But so far, I still cannot say it's which one is primary change. So it could be the secondary change as well. So right now, I'm still working on that, but definitely type 1 interferon signature higher in the ADA2 patient, I think could be true.
I have a question. So, Andre, you noted that there were basically two types of monocytes, the LGL monocyte, the monocytes derived from patients with the LGL phenotype and those with the vasculitis phenotype. And I was curious, Peter, if you looked at the differences in neutrophils in patients in those different patient subsets, and if netosis is different. And
no, we just purified monocyte because we had limited sample. So, but and we have also this model, so we can compare with healthy donors. But neutrophiles we haven't checked.
We haven't linked nets with clinical subsets in DADA2. We sort of took all comers. And I think at the time we did this, and correct me if I'm wrong, but a lot of the NIH cohort was more of the inflammatory vascular vasculitis phenotype, and so those are the ones that we primarily looked at.
Right, but now we seem to have more of the bone marrow failure
Yeah, it'd be
phenotype, so
interesting.
it would be interesting to see if it's similar or not.
Oh, the only thing that we know that AD2 binds to neutrophils, so that's why there is a link between this net formation and the absence of AD2, because and adenosine activates its net formation, so it's quite clear.
Can I ask Dr. Damwani, in your experiments with endothelial cells that express a lot of ADA2, have you also looked at ADA1?
Well, do they express it?
Yeah.
They do express it.
And in your experiments with knocking down ADA2, in all of those experiments, you were using siRNA to knock down.
In some, we also corroborated our findings with SH and CRISPR line, but most of the experiments are with SI.
So they would still, if they expressed ADA1, they would still be expressing ADA1.
Yes.
So how do you interpret results where you're adding exogenous adenosine or
So we don't add anything exogenously. Like
Oh, well, maybe I
we just
misunderstood.
supplemented the cells with nucleosides like adenosine and deoxyadenosine. So
But
they,
still, if you're knocking down ADA2 specifically,
okay.
and ADA1 is still there, it's going to
Yes.
affect the metabolism of those nucleosides.
But these are all relative experiments, so we compare it to controls, right? So you have control cells wherein you don't knock down ADA2, where and the other set will have ADA2 depletion in it, and you are making relative comparisons to control.
Yeah, I understand that, but you're still going to.
Yeah, definitely. We are not ruling out that there is no effect of ADA1, but there could be. But as far as our interpretations are concerned, we are very much tied in that.
All right. Oh, yes, please, Paul.
Just a couple of points. We've looked very hard for brain MVEC ADA2 expression, and we cannot find it at all. So I'm very interested in what you've just said, that your brain MVEC are expressing ADA2. We can't find it.
So it depends on what did you check, like which assay did you do? We did immunofluorescence and we did ELISA.
Yang?
Can you turn on your microphone,
Oh,
please?
sorry. We do both the Western and the qPCR.
So we do qPCR, ELISA, and immunofluorescence, and all three showed there is expression of ADA2 in brain and dermal endothelial cells.
But you talk about brain tissue or the brain vascular
Cells,
endothelial
primary
cell?
endothelial cells.
Okay.
Microvascular.
We don't.
The second point I was going to make is that it strikes me that this disease is a very good example of where we mean to move away from thinking about what's the best treatment for DADA2, that we need a much more precision medicine approach, and it might be that the first-line treatment for those with an inflammatory vasculitic phenotype, you'll use corticosteroids, which we're not hearing about, which still have a role, I think, and anti-TNF, taking away the corticosteroid over time. But for those other patients that have already failed anti-TNF, have a much more marrow failure phenotype, that medicines like baricitinib and JAK inhibition, if there's a positive interference score, could play a role. I'm mindful that the first indication for JAK inhibition was myelofibrosis, and that the end point of type 1 interferonopathies is marrow failure. I've had patients with CANDLE die with a fibrotic marrow. So I think that there could be a role indeed for a more precision for individual patients at that stage of the disease, different medicines, but always with a backup plan when things are not going well, that you've got the prospect of allogeneic hematopoietic stem cell transplant.
Yeah, and Paul, I think that raises a very important question, and that is really the ethics of when one can come in with an alternative treatment to steroids and TNF, because of, at least in the inflammatory and vascular phenotypes, the success of those treatments, one would, if you had a patient that was presenting to you at first, you probably would offer them steroids and TNF and anti-TNF, and I think that would be something that all patients, or nearly all patients, or their parents would choose. But there are settings, definitely, where one could consider alternative treatments, JAK inhibitors, or other kinds of treatments. Helen?
While we're on repurposing drugs, you mentioned from the endothelial point of view dipyridamole. I think that would make many of us nervous because of its antiplatelet agent actions, but of course there's theophylline. I can't remember my undergraduate pharmacology, but that too is a phosphodiesterase inhibitor and adenosine receptor blocker, isn't it?
So I can comment on this in this way. Like you see, DATA2 is being characterized with, multiple vascular clinical manifestations, new features, new phenotypes, as we discussed in session first. So it's possible that a subset of the patient which is not responding to TNF may respond to dipyridamole. So you don't know, because in some patients, maybe DPM won't be effective, TNF will be effective. So what I can say is that potentially single-cell RNA-seq analysis could be a potential and promising tool that can help us in understanding and classifying the patients in different subsets and see which therapies work in which subset of patients.
Rekha, when you said single-cell RNA analysis, what cells do you have in mind?
Well,
I mean, it's easy to sort,
yes,
peripheral blood leukocytes, but should you go or consider?
Well, again, I would say we can have easy access to monocytes, patient monocytes, and that would be the best model to start with.
Yes, Marco.
Question to Dr. Calvo, fantastic presentation. Thank you very much. Do you have the chance with these precious samples that you have to test and to screen for different cytokine expression in the bone marrow? TNF and,
we could actually
interferon type
start basically harvesting bone marrow supernatant and testing. That's a possibility, actually. That might be. That would probably be a good thing to do moving forward.
Yeah. Thank you.
I have a question for Michelle again. I think we sent you a different mutant that we were able to express. I think it was, is it the H112Q? Is that, do you remember? Well, the point is, I think we sent you another recombinant ADA2 mutant that didn't have ADA2 activity. Okay, because it would be interesting to test that to see if the nuclease is gone. And also, have you ever?
We have a mutant for AD2, which has no activity.
Yeah, we do too. And and we sent it to Michelle, so I was just curious.
Well, this kind of gets back to the question that we were discussing this morning, which is whether there is some other activity which is more relevant to thinking about the prognosis of the disease or the treatment of the disease. And so I do think that it's an important question of sorting that out in terms of different mutations and whether they uniformly reduce activity both of the nuclease and of the enzyme activity, or whether one, at least in some cases, can dissociate them.
Yeah, I have one other question about the lysosomal ADA activity. Well, there was a paper, and I think about this a while ago, from about 1993 by somebody named Friedman, I think, or Freeman, in which they purified ADA activity from lysosomes from both human cells and other non-human cells. And from the human cells anyway, they reported that there was ADA activity associated physically with lysosomes, and it was ADA1. And they would have seen ADA2 because they were using inhibitors. And I'm just wondering if you could comment on that.
Yeah, I remember that. I also read that article. But the ADA activity that they measured was, of course, located, I think, was it outside the lysosomes?
Well, that's the thing. You couldn't tell from the paper it was associated with the lysosomes. but I don't know that it was in the lysosomes.
My impression that it might be located outside the lysosomes, that it might bind to some protein associated with lysosomal membrane from the cytosolic site. Yes.
But they didn't find ADA2.
Which cells was it? Yes, that's very important.
No, I'd have to go back to the paper.
I don't think it was the macrophages. So.
Oh, maybe not. Maybe not.
Maybe
I don't know if it was brain, though. I mean, in other words, I just don't know.
Yeah.
It might have been fibroblasts.
They may have used cells that doesn't express ADA2, and that's, yeah.
Tara.
Hi, as a slight aside and a question to Peter Grayson, please. So we commonly use methotrexate in rheumatology, and we use it as a steroid-sparing drug. We've heard today it's been used to prevent a human infliximab antibodies in DADA2. Adenosine acts to increase, methotrexate acts to increase adenosine. That's one of its side effects. And we actually have an evolving literature that this may reduce cardiovascular events in our rheumatology patients. So I find it very interesting, especially as we use it in lupus as a backbone treatment for lupus as well, commonly use a drug that increases adenosine. We know that that happens. So how can we explain, by using methotrexate in patients with lupus, are we increasing their cardiovascular risk or problem, their netosis?
I would say that how methotrexate works is completely unknown.
But it does increase adenosine.
But it also has immunometabolism properties, which is where I think the cardiovascular benefit lies. We do have a person who's actually studying this in great detail who's in the audience if she wants to comment.
Looks like methotrexate had other mechanisms of action too, and we are looking into it. It's also activated an enzyme called AMPK, and like Peter was mentioning, it's unclear. Adenosine concentration is going up, but in a very, very low dose. Okay. And adenosine concentration is actually half-life is a few seconds. Then it's really hard to say that adenosine concentration is going to go up for a very long time, even in patients with methotrexate treatment.
Okay. Thank you.
Ivona?
I'm still thinking about this difference between patients that have severe inflammatory signature versus patients that don't have really that much inflammation. I don't know what is the most sensitive biomarker of disease activity, whether that could be, for example, interferon. I don't know whether anyone looked into interferon signature in patients that don't have, elevated CRP, but still end up with a bone marrow failure, So that's something that I don't know that anyone looked at. But now I'm thinking about Rekha presentation, about this endogenous retroviral element. And I'm just wondering, remembering there was a paper, and I don't know who published the paper, but I remember that there was a paper who reported a patient who became symptomatic after some viral infection. And now you talk about these endogenous retroviral elements. So is it possible to measure them, in peripheral blood DNA samples? Patient samples. Would it be possible to measure the expression of this? Because apparently if you have more of them, that's my understanding, then that would trigger interferon signature, right? At least in your system.
Well, I don't think that they will come up in a secreted form because they don't make the actual virus. It's just the element that they make. You don't get the actual virus. You just get their elements, DNA and RNA. So it's the real time is the only way that you can measure them. That's the only way to quantify them.
Well, it's also an interesting question. We heard earlier today about perhaps that there's some increased resistance to viral infection, actually, in people that have mutations in ADA2. And so it would be interesting to have more information in terms of possible flares of DADA2 associated with viral infection.
I want to comment Ivona's questions about this between two different phenotypes. I think the TNF-alpha itself could be one of the good marker to say between these two different phenotypes, because I did like TNF-alpha staining in both stroke-related patients and non-stroke-related patients, for example, just pure bone marrow failure. So basically, this with the stroke patients, they have much higher TNF-alpha signature for sure compared to the bone marrow failure patients, skin biopsy.
In what tissue? Where did you look for?
So far, for staining, I only did it in the skin biopsy,
biopsies, yes.
but we also did the iPS-derived myeloid cells with 3 different kind of phenotype. This is very preliminary, and one is just a stroke patient. Another one is like the stop codon pure knockout patient from Dr. Holland, and another one is the DAB patient. Basically, those stop codon patient and DAB patient, they don't have very high pro-inflammatory cytokines released with the myeloid cells, but in the stroke patient, they're relatively higher. So I think the cytokine itself could be one of the marker can distinguish both. Yeah.
And Kathy, have you done any immunostaining of bone marrow biopsies in these patients, in a sense that you can, for example, check the expression of certain cytokines in these bone marrows? I mean, you did a lot of immunophenotyping, but I just wonder whether you had a chance to look really how much inflammation is present in these samples, bone marrow samples, right? And then also whether you have, I know that this is not clinically indicated to do bone marrow in patients that have relatively quiet disease, but it would be interesting to know what's happening before and after the treatment. Do you see the resolution of these T-cell infiltrates, for example, whether
Yes.
you would see, decrease in the staining for TNF or possibly other cytokines, in these kind of experiments?
Yeah, we don't actually have antibodies in our lab for cytokines in the bone marrow. We could look into it, certainly. Definitely the patients that have been transplanted successfully, we do see obviously a marked recovery of marrow hematopoiesis and lack of T-cell infiltrates and T-cell aggregates. I think the suggested idea of isolating the bone marrow supernatant and doing cytokine assays would probably be a very good thing to do as well.
Mm-hmm.
But we can look into antibodies also.
You also talked about problem with B-cell maturation, and you also, I know that you've done now patients with SIFT, for example. So how comparable they are in terms of the defect that you see in bone marrow, in B-cell differentiation or maturation, actually, SIFT versus the other two? Where do you see more severe defect?
Actually, they're both, I guess, probably similar. Well, it depends on the patient, because you do have some data to patients who don't have the B lymphopenia, and we don't see the impaired B-cell maturation. So I would say, though, that the maturation defect looks similar. What the etiology is unclear. So it could be that you have the B-cell maturation problem because of the T cells. They could be, it could be immune dysregulation causing impaired B-cell maturation or destruction of mature forms of B cells, similar to what may be happening with the neutrophils. It's not clear.
Yes, Paul.
It strikes me from all of these conversations that the big clinical lead is for a biomarker that's not too invasive. And I think one of the key clues that we might have to study that is to focus on the pre-symptomatic patients. In other words, patients who have got the phenotype, they've got absence of the phenotype, but they've got absent enzyme activity and 2 mutations, but have absolutely no clinical features of the disease. And they've been picked up because there's an index case in the family. And if we can work out what's going on with them and follow them longitudinally, and are lucky enough even to see 1 or 2 patients switch into a phenotype and see what the difference is based on a variety of different biomarkers, that I think could provide a clue about what's going to be the best blood signature to differentiate that switch from inactive disease to a phenotype.
Unless it's a 2-hit phenomenon where you have to have some inciting stimulatory inflammatory event where adenosine goes up, and then the problem is in the response to that.
Almost certainly there will be a 2 or even 3-hit, intercurrent infection. There's all sorts of triggers that could be highly relevant in the pediatric population and also in young adults.
So Paul, I don't know, but it was related to this, sorry Gigi, but it was, I mean, the problem is that some DADA2 patients, I don't know, was my experience, do not have a chronic disease course. Those that have strokes, and some of them have the strokes when they have inflammation. So do you really prevent the treatment of patients that have no enzymatic activity? Because I would be very worried that when he has an infection or something like that, he can have some bad endothelial activation and strokes and so on.
I know it's a worry, but the first family that we started off with, this highly consanguineous family, the mother, who's in her 50s, is homozygous and has absent enzyme activity and is completely asymptomatic. And she happens to have four children, 2 of whom have had a severe-ish phenotype, and 2 who are completely normal, and yet have absent enzyme activity and the same homozygous mutation. So it's quite hard to persuade the mother, who herself has that same genotype and absent enzyme activity, no treatment all her life, that we should subject her children to immunosuppressive therapy for the rest of their life.
Yeah. That's why we do not reach the consensus in the because we think in a different way.
Okay, so Gigi.
It was simply a note in reply to Mike's comment about the 1993 Lindley's paper. I asked the best doctor after Wikipedia, which is Dr. Google. There was human fibroblast, so not sure it is relevant.
Yes.
Yeah, I just had a quick question for Dr. Calvo. I know all the marrow patients that have had hematological manifestations, and our 2 patients, the one that had pancytopenia, he had elevations in TNF-alpha on a cytokine panel. His brother, who had neutropenia, was like high normal TNF. I don't know if any of the other patients that you've looked at have looked at serum markers of cytokines. Have you noticed that even the patients that have pancytopenia, they have elevations in TNF-alpha or any other cytokines?
We'll have to go back and compare with the peripheral blood or the serum cytokine levels. That will be happening.
I'm curious about the interferonopathy and whether that has been only looked in the patients with vasculitis. Since at least in our patients with a cytopenia phenotype, they have viral infections. They are a problem to them. So with the increased interferon signaling, you would not expect to see that.
Yeah, so it may be that that's the basis of some sort of heterogeneity amongst the patients. That's right. Anyone have comments about that?
I mean, I don't have a general answer to this question. What I can say is about our cohort, and the proteomic is done on 6 patients. On the same patients, we tested the viral infection, same kind of cells. And all the 6 patients, they had interferon, with some more, some less. The homozygous splice side, so real loss of function, they had a really high amount of interferon. The compound heterozygous missense lower, for example, or homozygous missense lower interferon. So you still see the protein, but lower. But in all of them, you see an increase in interferon. Actually, if you remember the picture, there are also 2 healthy donors with lower level of ADA2 and increased interferon. I checked in the data sets, and the interferon signature is low. It's higher inside the cohort of the healthy donor. The 2 with the lower level of AD2, they have an increase in interferon pathways, not reaching the level of the AD2 patients, but yes. So it seems that there is this balance between, at least in our cohort.
And those patients, were they with the vasculitis, stroke phenotype?
Our patients, they are more hematologic. So our patients are more an immunodeficiency patient. So immunodeficiency, we identified many of them because they were hypogamma-globulinemic. So the first patient that we identified was hypogamma, was diagnosed as a CVID, but they have the same patient then in anamnesy had in the past also a stroke, but then not. Other 5 of them, they have a sign of bone marrow inflammation, but not a sign of vascular inflammation. And they are in general adult patients. Ours.
I have a question on the DNA's activity in terms of the lysosomal compartment. Do you think that DNA's activity is present even inside the cell, in the lysosome, or is it purely an activity that you see once it's secreted to the outside? Because it seems like the protein is collected on the outside and the activity is done on the outside. I guess that implication that in terms of a viral infection, when you acquire a virus, that gets taken up into the endosome or lysosome compartment.
Yeah, exactly. So the DNA's activity is associated with the lysosomes. It's not associated with the extracellular environments. And even if it's secreted, it has a maximum activity at pH 4.7. And at pH 7, it has no DNA's activity at all. So it's obviously an activity that is associated with the lysosomes. I just want to mention one more thing, because I'm working with these pig brains. And I have also found similar activities like ADA2, that means an acidic nicking endonuclease activity. So we may also talk about compensatory activities that can somehow affect or have some effect on the variation of clinical phenotypes. And you should also be aware that, I mean, rodents is quite close relatives to us. It's only 20 million years. But they don't have any ADA2 activity. And I think that shows that you must have some compensatory activities. And these activities I have found, and I try to determine what proteins they are. And maybe they can kind of explain both the kind of mild phenotype of DADA2 and also the difference in the clinical phenotypes.
So are you talking about other nucleases, DNases that are associated with lysosomes other than that with ADA2?
I don't know if it's they're associated with lysosomes. The reason that I say it is because they have a similar pH optimum, which is around 4 to 5. So I assume that they are associated also with the lysosomes. And that comes into the next, it's starting to become a lot of experiments to do here, because you are now talking about maybe a pathway for DNA degradation, which is not limited to the DNase 2, but you have nicking endonuclease. And just recently in the Nature Immunology, it was also written about another enzyme recently discovered, is a lysosomal exonuclease, which cleaves one-stranded DNA. It's also a group from La Jolla. And that was actually two enzymes. That was they made some knockout mice. And phospholipase D3, I think it was associated also with myeloid cells. And they caused a mild autoinflammatory disease. But if they knocked out another enzyme also, which had a similar activity in phospholipase D3, they get a strong interferonopathy. So it may be that products of ADA2 is actually connected to a pathway where also these exonucleases are involved. And now we see contours of a DNA degradation pathway. And also kind of a very close relation to autoinflammatory diseases.
Can I ask if you, or Michelle, maybe, do you think there's a way of looking for the substrate of the ADA2 nuclease activity that accumulates in cells of, that are deficient in DADA2? Or maybe challenge them with something that would cause it to accumulate?
I mean, there is a possibility. I see it technically super tricky because you should select the right cells in the right. Because, for example, looking at the physiopathology, I would really like to see in those tingible macrophages which kind of DNA is accumulating with or without ADA2. And then sequencing, for example, you can imagine to a chip sequencing or a next generation sequencing. You can precipitate the ADA2 and to sequence what is there to have an idea which kind, if it's like sequence specific or whatever. But this technically is quite difficult. I mean, I can imagine to, in terms of experimentally what to do. I cannot predict how much and if it can be really done. So technically, I see it quite tricky, let's say.
Well, one way might be, so in ADA1 deficiency, I hate to come back to it, but the problem is really a ton of deoxyadenosine that is generated from apoptotic cells in environments where you find them, like the bone marrow for erythroblast nuclei that are lost, or proerythroblast, and in the thymus from negative selection. So I wonder if you took macrophages from normal and ADA2 deficient patients and fed them apoptotic cells.
This is exactly an experiment that we plan to do, to take in ADA2 monocytes, differentiated macrophages, and to co-incubate with apoptotic cells. And then to even sequencing the DNA or just measure the concentration of DNA via qPCR, yeah. Or just measuring interferon, for example. Yeah, definitely.
Yeah, for this question, I think I did some adenosine measurement in cells, and I think we see some change, but not very dramatic. For example, in the stop codon patient, that's specific ADA2 deficient cells, completely deficient, and that one has a little bit like difference with control. But the DADA2 patients, those cells, we use, but we didn't see difference. Use the iPS-derived macrophages. We didn't see difference. So they have slightly changed with the homozygous knockout, but not with this.
Okay, Troy.
I was just going to ask, so we've been talking about deficiency, but when you think about replacement, I mean, there is, you mentioned the mouse or the fact that rodents, Ola Christian, that they don't have CRC or their AD2, and but there is a group that's made and published transgenic, where they put it under either driven by a beta-actin promoter or driven by a myosin heavy chain promoter, that the mice actually were not normal. In fact, some of them were embryonic lethal. They had significant cardiac hypertrophy. Some of them were missing, eyeballs. Some of them had kidney problems. So I guess in the setting of the mechanistic, I guess ask everyone on the panel who's been studying this mechanistically, if you sort of flip this around the other way and based on the mechanisms that you've hypothesized, if you think about overexpression, if you give too much ADA2, which is, of course, has implications for treatment, do you see a mechanism whereby you could get this sort of a phenotype of cardiac hypertrophy, things like that? I mean, the beautiful thing about ADA2 in humans is that it's very tightly regulated, and it gets upregulated in inflammation and goes back down. And maybe there's a reason for that, which may be important when we start thinking about treating this disease. So anyway, I just wanted to get your thoughts.
I can only talk to you about it in a lysosomal way. So if you look at the lysosomes, I think that when concerning the DNA degradation, it's not sufficient that the DNA is degraded, but it's also important that the fragments that are produced can interact with the DNA sensing apparatus like the toll-like receptor 9 or a cytosolic DNA sensing protein. So I can just imagine if you underproduce it, of course you will get different kind of DNA fragments, and that can cause interferon upregulation. And I can also at least think conceptually that if you overproduce the enzyme, it may also cause some kind of changes in the fragments that you are getting, and can also cause some kind of a disease phenotype. But of course, you're talking about something that I don't know so much about, but I can think that it can have some.
I mean, if we're going to
some
speculate wildly, another possibility is Cardiac myocytes are very enriched in adenosine receptors, and it's very important to cardiac physiology. So perhaps ADA2 can bind directly to adenosine subtype receptors and modulate preferential signaling through certain ones, and resulting in those types of phenotype.
Let me just comment on the lack of any cardiac abnormalities in kids with SCID due to ADA1 deficiency who have a lot of adenosine.
All right, well, I think that at this point, we're ready for a break.